Okay, so short. Okay, okay. Good morning, everyone. So I'm Satoshi Maeda of Hokkaido University, the chair of this session. It is my great honor to chair this session. We have two speakers and by the two distinguished invited speakers. Time is limited, so I move to the first lecture. It is by Professor Thierry LANGER. Maybe Thierry is very famous in this community, but let me introduce him very briefly. So he got a PhD at the University of Vienna, and in 2003, he founded the company Intelligando GmbH. And in 2008, he was appointed CEO of Prestwick Chemical Inc. And under his leadership, several drug discovery programs in different research target sectors successfully progressed into preclinical and clinical development. In 2013, he was nominated full professor for pharmaceutical chemistry at the University of Vienna, Austria. His research interest ranges from medicinal chemistry, computer-assisted molecular design to algorithm and software development, and his expertise and scientific work have culminated in more than 250 original articles, book chapters, and invited reviews, several patents, and more than 350 lectures and poster presentations at scientific meetings. Okay, so please.
Thank you, Mede-sensei, for this very kind introduction. So I want to thank especially Sasha Warnek and his team to put this fantastic summer school again and again. And it's really a pleasure to be here back in Strasbourg. I have been living here almost 10 years. I have very good memories. And I want to thank you for being here at 9 o'clock in the morning after this very nice evening that we had all together. So you might think that pharmacophores are old-fashioned, but no, I will show you why. But first, I want to acknowledge my group here in Vienna, but not only people in Vienna. So a couple of people are here from Vienna. A guy from my group, Daniel Rose, I will highlight his work later on. Of course, I have also to acknowledge a lot of work from people that were at the University of Innsbruck. Before I was appointed CEO of Prestwick, I was in academia in Innsbruck and had a very nice group there with seminar contributions from, for instance, Johannes Kirchmeier, Daniela Schuster, Christian Lagner. Then, as Mede-sensei mentioned, I founded this company together with two colleagues. And after I finished my time here in Strasbourg, I went to Vienna 10 years ago, and I was extremely lucky to attract again very, very talented people. So all of this work is not my work, but it's work of these people. And I'm really, really happy to have them around me. And of course, it's also necessary to have some funding. We are very well funded, both European community and some private public partnerships. And Intelligand, of course, is the company that takes the algorithms that we develop in our group and transforms this into professional, highly stable software. Of course, what we are talking about at the very early stage in drug discovery is the most crucial question is to find good starting points, right? To find good hits, to develop them into leads, and to optimize these leads to clinical candidates. It means to find the needle in the haystack. And I still remember a person many, many years ago, Roger Lahana, who made this quote and said, finding a needle in a haystack, maybe it's not a good strategy to make the haystack bigger. So now we see that the haystack is made bigger and bigger and bigger, but I'm not sure if this idea of Roger Lahana is still valid. It could be that we find better hits when we make the haystack bigger, and we will see. So some important questions here that we have to tackle is about data analysis and data representation. Right? Representation of data is as equally important as the analysis, because you need to take use of this data to make better decisions. Right? So this is extremely important. And I'm convinced that pharmacophore models are still a very valid way of representing data, especially if you want to translate back and forth from the synthesis to the design and from biological assay back to design and synthesis, because it's not a black box. You can see why a molecule is better than another one. And therefore, we are still working on these methods that we did not invent, but we contributed a lot to the further development. So understanding molecular similarity, for instance, right? You can compare compounds structures in view of their preferences for specific molecular interactions, the interactome. Remember the lecture of Gisbert Schneider yesterday, talking about the interactome, how compounds interact with biological targets. So a way to do this is annotate all pharmacophore features that a molecule has or could have, and then look at them at those who are really the important ones. And you can do this, for instance, very, very easily with a click of a button if you have a cryo-EM structure or a X-ray structure of a ligand in a protein, right? So you can then find exactly which are the compound parts, the pharmacophore parts of the molecule that are important for interactions. And this is one example that I took out of a PhD thesis of Doris Schütz here, who did her PhD in Vienna in a consortium, in an IMI consortium. And she was looking at HSP90 molecules, and she was very much interested not only into the static, but also the dynamic behavior to understand kinetics, K on, K off. So what we can do now is we can translate MD simulations into multiple pharmacophores and understand the binding kinetics in a pharmacophore description and take our ideas for design from these trajectories. So this is a video here that is a little bit shaky because it's Windows, so on my Mac it runs much smoother. But you understand the idea, right? You see the pharmacophore moving in the trajectory. You can analyze, in our software, you can analyze the feature frequency, how many times a feature really occurs. So you see here the pharmacophore features in a time diagram. You can understand which feature, which models that you record during the trajectory have specific features. If this amino acid is contributing all the time with the specific interactions to the ligand binding or only rarely. And you can even see here in a percentage of how many times, how many frames in your MD simulations you will have a specific interaction with a specific part of the molecule. So this is extremely useful when you do design and optimization of molecules. Of course, it's not really something that you would do when you start a screening campaign. So I want to come back here because this person, Camille Vermouth, whom I made postdoc with in 1991, 1992 here in Strasbourg, he was the guy who had introduced computer-aided molecular design in academia. He was the first one who had the software from Tripos Sibyl at that time in his lab, and people from industry would come, the lab would be closed down, everybody had to leave, they would work, and one day later they would go back again to their company with the tapes and everything. And he had all the things in his brain. So he used pharmacophores every day to explain to his chemists how they should modify their molecules. And this was extremely inspiring. This is the definition that Camille Vermouth set up for the pharmacophores when he was the president of the MedChem section of UPAC. And this is really the meaning of a pharmacophore. It's not that you have one group that is important for the activity, it's the totality of all groups, right? And you can look at this a little bit in a more abstract way as saying the totality of universal chemical features that represent the specific binding mode of a ligand to a molecular target. So it's the interactome, by the way. And these interactions are electrostatic, H bonding, aromatic interactions, hydrophobic regions, coordination to metal ions. And you can go from 3D down to 2D even and explain this where people talk to each other. Medicinal chemists, especially the older ones, don't like too much to look at 3D structures. The younger ones love 3D structures. So they are used to this, but you can do everything here. So, as I said, Pharmacophore model is the most suitable data representation method for guiding medicinal chemistry. Is it also useful for finding hits? I believe so. So I just want to touch really briefly on Ligand Scout, which is the platform that Intelligent has been developing since 20 years now. So it's a fully integrated molecular design package with high-end graphical user interface. We have workflow integrations in 9. It has a worldwide user basis. It's, in our eyes, the leading solution for pharmacophore-based modeling. And we are developing actively next-generation capabilities. And now, since 2006, when we first introduced the product on the market, there are more than 3,300 publications that have been using Ligand Scout for identifying hits, for repurposing molecules, etc., etc. So you can look this up in Google Scholar. You will find a lot of interesting information. And one recent success story from Pfizer I want to mention here, and we know the guys who did this. They found in their internal library with Ligand Scout a molecule that they did not find with any other virtual screening software. So they had almost given up on that. The hit that they found was very potent. It was already 17 nanomolar from their own library. So nobody has access to this molecule but Pfizer. And then they did a rapid hit to lead. And this was the lead here. They have not yet published a clinical candidate, but soon they will publish it, they told us. And just adding a chlorine atom here on the initial hit, you get already almost a factor of 10 better. So extremely fast, extremely high quality lead. And the other thing, I still believe that generative modeling, making molecules, rather than looking into gigantic screening libraries, is a good strategy to find interesting structures. And here, this is a paper from Japanese colleagues who designed and synthesized DDR1 inhibitors by using a generative network that builds molecules pre-trained on Campbell. And the scoring function was the Pharmacophore fit score. So after a while, the network learns how to make molecules that fits the Pharmacophore. And then they selected some 10,000 molecules, recalculated the binding affinities, synthesized them. Most of them were active. This was the most interesting one here in the dual-digit nanomolar range, new molecule designed by a regenerative neural network. So I think this is really very interesting. And I just want to tell you what we are doing now. So we are just also employing neural networks, machine learning model for chemoinformatics. We have just tapped into this a few years ago, and people in my group were really interested in this. We have built some new algorithms. We have built quantitative pharmacophore models, and at the end, I will show you some next generation descriptors for pharmacophore modeling. Again, this whole thing is developed, put into a toolkit that is freely available. I will show you the link to this in the next slide. But also, when the algorithms are really mature and well validated, they are re-implemented on a professional basis into Ligand Scout, and we will call this Ligand Scout XT. So this is the toolkit. So take a picture of this QR code if you want to use it. I know a lot of people use it already here in the room. It's the CDP kit. You can also see here cdpkit.org. So it's a toolkit that is developed mainly by Thomas Seidel and Oliver Wieder in my group, and implemented in C++, has all basic functionality for chemoinformatics workflows, has several binaries that you can use directly, and comprises this chemical data processing library that we call CDPL, together with some command line software tools, and has an API to C++ and Python, and contains the major machine learning libraries such as scikit-learn, PyTorch, and TensorFlow. So you can, based on this toolkit, you can build up your own workflows. And what I said, so it's really our standard environment for prototyping novel algorithms and then follow-up implementation to Ligand Scout. So a few examples that I will show you. A new graph neural network architecture that we developed for better predictions of log P, log S, and log D, for instance. A new high-performance conformal generator that is openly available and also implemented in LiganScout XT. A new alignment algorithm, novel descriptors for Pharmacophore modeling. And we are also extending Pharmacophore capabilities to the exascale. Now, exascale is not 10 to the 40, but we can screen conveniently 10 to the 10 now. And we see all the molecules. And this is really a great thing. So one paper here that was a new combination of neural networks to address this difficult question of predicting, improved predicting lipophilicity and aqueous solubility. Combines actually a directed edge graph isomorphism network with a direct message parsing neural network. And we could show that using this combination of two neural networks after each other could drastically improve the prediction capacity of our system here. And we successfully used it for predicting log P, log S, and log D. So you can find this, and it's all available also online. Another algorithm that was developed in our group is an improvement of the existing pattern matching-based algorithm that makes LiganScout so fast. And actually, the more features you have, the faster it gets, right? If you have 15 features, it will screen like this because it can throw away a lot of compounds before. So this new algorithm here addresses a small issue that we had here when in the pattern matching algorithm, when we were aligning perfectly 3 of the pharmacophores, sometimes one feature could be outside and then would not be recognized as a hit. And when you look at this would be the desired alignment. So we have built this on by using heuristic guesses at the beginning to very quickly go into plausibility alignments and omit those that are not successful. Also, we have been able to address the problem when an atom lies at the edge of an exclusion volume, for instance, that would invalidate the alignment. We can redo the alignment and modify it a little bit so that still this hit would be found. And the big thing that is the big advantage over the old algorithm is that it does not slow down if you have omitted features. So you can have a new use case, for instance, you generate in an ApoCite a pharmacophore model containing 30 features or 40 features. And then in the screening, you can ask to have a minimum, for instance, of 8 or 9 features. And the algorithm will be almost as fast as if you would ask for a complete match. So this makes now a sort of pharmacophore-based docking completely accessible. And I'm very excited about this. So this is how we do it. So we can really have a more accurate retrieval according to the user query. And it's optimized for a maximum number of matched features, adapts also to exclusion volumes. And we can tune for runtime and accuracy. So if you screen now in the exascale, you will be a little less accurate and faster. And if you screen your own library in-house, you want to have highest accuracy, and then you're a little bit slower. But you don't want to miss hits, right? And this is given here. The next question is, of course, how to rank pharmacophore features. And I will not touch on the structure-based modeling now. I will touch on the ligand-based modeling. So if you have data sets of ligands together with bioactivity values, you might want to make quantitative models. And there were methods available, for instance, in Catalyst, now in Discovery Studio, but these methods are limited to a low number of features. So the hypogen method of Catalyst, for instance, stops at 5 features. So you cannot have more than 5 features in your model. Makes no sense because the more features you have, the better you can describe your chemical space, right? So we have published and elaborated an algorithm that is also available in the toolkit, which is called QFAR. It's a quantitative pharmacophore activity relationship method by Stefan Kohlbacher. And the idea is to take a training library to select a template, which would be the most rigid molecule with the conformations that this rigid molecule can do. Then in a second step, align all pharmacophores from all conformations for the entire training set, cluster them, have a container, post-process the thing, and then correlate these aligned samples using a machine learning method that trains the feature weights according to the binding affinity or Ki or whatever you want to predict here. And what we used here is a random forest model that turned out to be the best method that we could use. And we can use this with a small number of compounds. Decision trees are not relying on huge data. So you can really do this pharmacophore modeling with 20, 25, 30 molecules, right, already, and get something. So this is available. Then we have used this for predicting quantitative neurotoxic effects. At the end of my lecture, I will give you the frame where we did this. And even then put this, so this is, as you see on GitHub here, Q for applications. So this is a fully automated way now you can do this. And we have also implemented this method into KNIME nodes. So if you use KNIME, you can use this here to generate your models, to read in the models, and to predict outside data sets. So just go there and check for that. So now let's go back to the haystack. Bigger haystack, better hits. This is the question. So is it true? So there are articles that claim, and there's one paper from Brian Scheuket, who clearly says the more molecules you start with, the better will be your hits. Well, this is one opinion, right? But for sure, you will have more hits, right? So if your method retrieves X hits in Y molecules, if you multiply Y by a factor of 1,000, you will also multiply your number of hits, right? So the question is, which size is ideal for hit identification? Please don't believe that you can find the drug in the haystack. You will find hits. You will never find the drug unless you can enumerate 10 to the 120 molecules, which is absolutely impossible, right? So you will never find the drug. But you will find probably good hits. Now the question is, how many good hits can you manage, right? So we have done this experiment together with a company partner that we developed this method with, and we screen now between 1 billion and 10 billion molecules. So if you have a pharmacophore that has 10 features, you get already quite a large number of hits. So I would say, if you are able to retrieve out of this 1 to 10 billion compounds a size of maybe 1,000 molecules, this is still something you can manage. And then you can select maybe 500 from real space and buy these 500 molecules and test them. And then you still probably have problems because you will probably have a very high active hit rate. And then you are really stuck with which series should you continue to develop. But this is a good problem, right? It's not a bad problem, at least. So I believe that screening between 1 billion and 10 billion of a good, well-diverse selected set with a precise method will give you good starting points. But for this, of course, you need still methods where you can enumerate the conformations, because we are still working in 3D alignment. So we also reworked this problem. We had pretty good conformer generators already, but we made a better one. And this conformer generator that Thomas Seidel developed together with Christian Perman, Oliver Wieder, Stefan Kohlbacher, it's twice as fast as the fastest before. And again, also this one is available in the toolkit, in the CDP kit, so you can install this conformer generator. And Thomas has benchmarked this, Thomas and Christian have benchmarked this, and it's the best non-commercial conformer generator that is out there at the moment. So now we're integrating this into Ligand Scout XT to get first of all, faster database generation of these billion-sized libraries, right? And also, we reduced the database size format in comparison to the previous one. So the previous one was the Ligand Scout database format LDB file. 1 billion compounds currently take up 16 terabytes. So this is still something that is manageable. And together with this G3PS alignment that I showed you, it forms the basis for Pharmacophore-based exascale virtual screening. And it works already. But isn't there a better way to pre-filter? And this is where I want to show you some work that has been done by Daniel Rose. Daniel is here in the audience over there, and he has a poster. So if you are interested to discuss more about the details, please visit his poster and talk to him. I just briefly will introduce you to the idea behind this. So this is the traditional way. Yeah? You have your ligand database with conformational database. You have, for instance, a PDB structure. You generate a structure-based model, or you can generate ligand-based model. And you filter this with alignment. You get your hit list. But this 3D alignment is still costly, right? It's, even if we made it extremely fast, it's still limited in the speed when you go to the very large scale. So the idea that Daniel had to use a new concept, namely contrasting, to build a contrasting learning framework to map pharmacophore models into an order embedding space and using vectors instead of pharmacophores so that you can omit the alignment. So the idea is to encode the conformational database into vector representations and also to encode the model. And then in this embedding space property, look for which vector here, or this vector here, where is it found here? And you get your hit list. It's less precise, of course, than the alignment, but it could be a very wonderful pre-filter tool to go down for hundreds of billions to an amendable number for accurate screening. So just a few slides here that I borrowed from Daniel to explain a little bit this vector embedding space. So these vectors here in this example here, you have these vectors, for instance, and you see computer science, data science are vectors that really point in the same direction because the embedding is showing the same thing, right? So it's concepts. Also, for instance, here caffeine and molecule should also point in the same way because caffeine is a molecule. So then if you look at molecules and data science, right, chemoinformatics should be somewhere in between. So this is a way to look at the properties of these vectors showing different concepts. And so this is now it's called this retrieval of concepts in databases or in information. This representation can be used in search engines, it's used in recommender systems, and it's used also in chatbots like ChatGPT are using exactly this system. So now the question is, how can we encode these pharmacophores into vectors into a way that the system learns a pharmacophore in this embedding space? And Daniel came up with a fantastic idea to use a Siamese network and use contrastive loss function to adjust the weights to kind of embed the vectors in the good space, in the right space. And so the question is, what shall we compare to train a pharmacophore embedding model? And here comes the concept of subgraph and supergraphs into play here. And a supergraph here, for instance, that could be, or a subgraph that could be a pharmacophore model here. A query pharmacophore model could be found in a supergraph. And we just need to use the vectors and the same concept. And there is a concept for this that has been presented by a Chinese colleague, by Ying, that I will show you on the next slide. But he has shown that if you do a principal component analysis of these first two embedding dimensions, you can already see where your target region would be. So if your query embedding would be here, for instance, the target region would be here. And you would only have to look into these pharmacophore vectors that you have and not here, if you have, for instance, some off-target embedding. So it's subgraph, supergraph isomorphism that is used here. And so what we do is we can train a positive pair that is a subgraph of the supergraph in this shared weight Siamese network and derive these shared weights using this contrastive loss function. So this is a fantastic way how Daniel transformed this concept into the pharmacophore space. And the pharmacophore space, again, has the advantage that it's not about atoms, but it's about interactions. And you don't care about the atoms, you care about the interactions, right? This is the thing here. So, this algorithm by Ying is the neural subgraph matching. Unfortunately, the paper that he submitted and that Daniel found in the preprint archive was not accepted for publication because of some reason that the reviewers didn't like, but Daniel adapted this to this pharmacophore problem, and it's really working well. And so we could show in a benchmark data set here that we took the benchmark data set from Didier Rognan and use an arbitrary query pharmacophore, and these are drug-like ligand database. And what is shown here in this space here is all the pharmacophore vectors here in the first and second principal component. And you see here the color here encodes the number of features. So you see here in the target region that is defined here because the subgraph is here. So you only find pharmacophores that have a high number of vectors that have a high number of features. And so the interesting thing is here we compare now the screening time. If this is a small data set, like almost 4,000 ligands. In the CDP kit, it takes 2 seconds for screening with the traditional methods. So it's still fast, but it's only 4,000 molecules. But in this, what Daniel called vector match or pharmaco match, we still are discussing about the name. It's 100 times faster. It's only 0.02 seconds. And we looked at the true hits, and there's a big overlap between the two methods. And there's even ideas to go faster, to use specialized vector databases instead of a normal database that is used now. So a vector database could be used for faster retrieval because we could exploit the inherent tree structure. So we go here to the entry point, get to the next one, go to the next layer, transfer to the next entry point, and so on, and look always at the nearest neighbor. So this would be a method where you really could speed up this vector search, probably by a factor of several orders of magnitude. And there's another application for these pharmacophore vectors. The big, I would say, one of the weak points is entropic contributions of pharmacophores because of the placement of the hydrophobicity features, right? So we are at the moment, we are still working on this method where we place hydrophobic features onto a molecule where hydrophobic regions are that are surface accessible. But, and this, we have published this more than 10 years ago. But a solution could be, rather than put classical hydrophobic features onto a molecule, to take atom-typed hydrophobic features and use a point cloud encoder to again do this vector embedding. So we could then have a much improved description of hydrophobicity and probably overcome this problem of the entropic terms not being so well explained with pharmacophore methods. I'm very optimistic, and we will still see how this works. So now let's just touch a completely different topic here. I showed you some very exciting new applications from our lab. Now I want to show you in the last 5-6 minutes of my talk how we use these models in a big collaborative effort to de-risk molecules for neurotoxic outcomes. And there is a poster by Sharon Bryant and myself also on this This was a project that now is finished. It's the NeuroDerisk project that obtained funding from the IMI, Innovative Medicine Initiative. Sanofi was the project leader. I was the project coordinator, and there were like 5 big pharma companies contributing data. There were small companies, and there were also a lot of academic groups building methods to find out if compounds might have a risk of neurotoxicity. And this is still something that is not addressed, and a big problem, especially in late clinical stage. So there's the website, neuroderisk.eu, where you can find everything. And what we were aiming at developing predictive models, in silico models. This was our part. Of course, there were also biomarkers identified, and there were in vivo studies done also to validate the models. So this was a whole consortium of more than 80 people working together. Our job was to build neurotoxicophores, qualitative and quantitative ones, for seizure risks. So a lot of compounds induce seizures. And we know why, because they bind to GABA receptor. Then also for psychological misbehavior, aggression, suicidality, this is a big problem. And for PNS, namely peripheral neuropathies, that are very much endowed if you use, for instance, if you have cancer and you use anti-cancer drugs. Also, very often you will have neuropathies and pain in your legs and hands and so on. So for all of these 3 different areas, we develop neurotoxicophores. And there is ways, of course, we can do a target-based approach if we know exactly what the target is. And this was for seizure, for instance. So here we know, we look at these properties. There are even co-crystallized structures or cryo-EM structures where we directly can derive such a model, train it. But also there are things like suicidal ideation where we don't know what the mechanism is. But luckily in the project, we had a company, Alcediac in Montpellier, who had tested all drugs that are known to be suicidal. And they have identified biomarkers. There is a mechanism for that. It's basically, it's a modification, post-translational modification of serotonin receptors that make the receptor has less affinity to your own serotonin, and then you kill yourself. So this is the target-based approach. No question. I mean, we can build these models, we can validate, we know exactly how it works. In this suicidal ideation problem, so what we had here, this was this 1,492 drugs that have suicide annotations from FAERS, from MetaADB, and NIH databases. So we did a lot of models here. We created more than 45 ligand-based models. So you don't have the target, you have ligand, you have effects. So we built ligand-based models. And then we tested these models on the data set of Alcediac. And we retained only the models and put them later on in the toolbox who were predicting correctly. So if you're interested in this, it's EDITOX. And this is an unambiguous test here that is really showing if a drug in a patient will induce this or not. And it's a biomarker that they can measure now, and we can predict it. So what it's built into a NIME platform, actually. So these are the models here, GABA models, PNS models, suicidal ideation models, and then we have the NeuroDerisk profiler that has all the technology built in from conformal generation, pharmacophore-based alignment, output, etc. And you get this kind of heat maps here. You can link this in the NIME platform here to a database, or you can draw your molecules, or you can read in Excel sheets or whatever. And then all the things will be done here. There will be conformers will be generated and aligned to the pharmacophores. And then you get this heat map with all the models. And if you have a red or orange or even yellow flag, you should take care and go into more detail. This is now currently used already in several pharmaceutical companies in the compound registration system. So every chemist that gets his compound into the system gets at least a flag if there is some problem, and could think about how to modify the molecule to get this flag off, right? So with this, I want to end, and I want to give you some take-home messages here. So I really believe that these next generation pharmacophore models, methods are ready for the most difficult challenges that we have in computer-aided molecular design, especially in this hit ID, hit to lead, and lead optimization. They are highly useful for hit ID and prioritization. They are amendable for high precision virtual screening in exascale libraries already, so we do this. And they are most comprehensive for medicinal chemistry guidance. Because you talk to a medicinal chemist and you tell him, please put an H bond acceptor there and a hydrophobic feature there, and then you will maybe avoid a target, right? This is also a good idea, not only optimize for one target, but optimize for selectivity and get rid of off-targets. It's as important. And you can take into account dynamic effects, binding kinetics, for instance, and translate them directly to design when you use these dynamic pharmacophores. And I believe that in the future, we will have better lead structures that are void of toxicity risk because of this method. So this is very optimistic. I invite you to visit the website of my group here in Vienna, Chemoinfo. And if you are interested, of course, visit the website of the company. And I'm happy to take your questions. Thank you very much. Thank you.
So I can ask a couple of questions. So first,
yes. No, I can hear you. Ah, yes, maybe on the Zoom, maybe give him the microphone so that. Or I can repeat the question.
So thank you for this insightful presentation. I was just wondering, because the constructive learning that you presented actually is quite challenging, because you have to actually choose the negative samples. So you built a really good embedding model. But actually, have you tried to instead combine your like your previous approach, like QFAR, with constructive learning, so that you don't need to really choose so much negative samples as from the properties, we can have the same signal?
Yeah, no, this is a good idea. Yes, we have not tried this yet. We have not tried this. This vector representation is basically could be also very well adapted to these quantitative models. Yes, yes. And Daniel will probably go on in this direction. So there are so many ideas. We really have to categorize and do the most important things. But I think this is a very good idea. In the pharmacophore vector, these negative things we can do then by modifying a little bit the positions of the pharmacophore features, and then have this. But go into more details with Daniel. So Dragos had a question. Yes.
Actually, I already asked it to Daniel yesterday.
Okay.
Embedding is nice and sexy, but you don't need embedding to make a vector out of pharmacophore. You know, what about good old pharmacophore colored autocorrelograms or triplet counts? Do you have any positive proof that embedding is better, or
well, I
can
think
use a Tversky metric with those good old descriptors and see what happens?
So what did he tell you? He is the expert. I'm not the expert in this. I see your point. I see your point. Actually, I do not have really the answer for that, but we can discuss the three of us. Yeah.
Okay, so yeah, thanks, and the
Thank you. Thank you.